Hello dear student, welcome back again in this lecture video. I am Pratibha Bhagat and today I will discuss about another field crop disease, that is cotton. Cotton is a very important fiber crop in India and also in world, which is affected by numerous pathogens, but according to your course diseases of field and horticultural crops and their management part 2 syllabus, we have to go through only wilt, anthracnose and black arm, and these following diseases are very important and very dangerous in cotton. They can affect the directly the yield of cotton. And so let's start from the wilt, which is caused by a fungus, and that fungus is Fusarium oxysporum F species vs infectum. एंड यह डिसीज़ इंडिया में वैसे हर उस जगह पे देखने को मिल सकती है जहाँ जहाँ पे कॉटन का प्रोडक्शन किया जाता है, बट इंडिया में इस डिसीज़ को सबसे पहले नागपुर में 1908 में रिपोर्ट किया गया था। एंड साथ ही साथ ये डिसीज़ हैवी सॉइल में बहुत ही सीवियरली अकर होती है, लेकिन लोमी सॉइल में इस डिसीज़ का इंसिडेंस कम देखा गया है। सो लेट्स स्टार्ट फ्रॉम द सिम्टम्स. So cotton this disease can affect the all stages of cotton, and the earliest symptoms appear on the seedling in the cotyledons, which turn yellow and then brown. The base of petiole show browning followed by wilting and drying of the seedlings. In young and grown up plants, the first symptom is yellowing of edges of leaf and area around the veins, that is discoloration start from the margin and spread towards the midrib. The leaf lose their turgidity, gradually turn brown and droop, and then finally drop off. Symptoms start from the older leaves at the base, followed by younger one towards the top, finally involving the branches and the whole plant. The defoliation or wilting may be complete, leaving the stem alone standing in the field. Sometimes partial wilting also occur, where in only one portion of the plant is affected and the other remaining free from the symptoms. The taproot is usually stunted with less abundant laterals, and browning or blackening of vascular tissue is the other important symptoms. Streak or strips may be seen extending upwards to the branches and downwards to the lateral roots. In severe cases, discoloration may extend throughout the plant, started from roots extending to the stem, leaves, and even to the balls. You can see in this image. You can see the partial wilting symptom. Few leaves are still alive, and the few leaves are dead because of the partial wilt. And if we go to the next image, you can see there is a sorry, there is a black to brown color vascular tissue is affected by pathogen, and you can see the brown or black color strips or streaks inside of the main stem. This is also a very important symptoms of the fusarium wilt. And let's go, the next is pathogen, means etiology, how fusarium oxysporum looks and how it appears on the plant. So as in this image you can see, this is the pathogen of wilt. And as a taxonomical tree, if we go through the taxonomical tree, this pathogen belongs from the subdivision Deuteromycotina, and the fungus produce microconidia and chlamydospore. The first one, which is very long, that is macroconidia, and this small one is microconidia, and this one is chlamydospore. So microconidia are means macroconidia means big conidia, that's why it's called macroconidia. So microconidia are 1 to 5 septate, as you can see in the picture, and it is hyaline, thin walled and falcate with tapering ends. And the second one is microconidia. Microconidia means small conidia, small size. Then the microconidia are also hyaline, thin-walled and spherical or elliptical in shape, and they are single or two-shelled, but they are not septate. Then if we go through the, if we can see the chlamydospore are dark colored and they are thick wall. Usually they also they also come in intercalary, and they can also appear as terminal chlamydospore. So this was the structure of pathogen Fusarium oxysporum F species vaccinfectum. Then next is disease cycle and favorable condition. So this fungus can survive in soil as saprophyte for many years, and chlamydospores act as a resting spores. The pathogen is both externally and internally seed-borne. Mainly, the primary infection is start from dormant hyphae and chlamydospore in the soil, and the secondary spread is through conidia and the chlamydospore, which are disseminated by irrigation water. Means pathogen firstly introduced in the field by the dormant hyphae or chlamydospore, which is present in soil. But this disease also seed-borne pathogen, and the secondary spread uh, can be start from the choroidea or by chlamydospore, which are disseminated, means which are spread by the irrigation water. This was the disease cycle, and these are the disease cycle for this disease. You can draw this image while you're in your exam also, or in your practical files also. So next is favorable condition. This disease, various factor are favors the deceased for its development. First one is soil temperature. Soil temperature of 20 to 30 degrees Celsius is very favor the disease to its development, for its development. And the next one is hot and dry period followed by rains. Third one is heavy black soil with alkaline reaction, and then also increased doses of nitrogen as phosphatic fertilizer also increase the incidence of the disease. And then wound caused by nematode and grubs will also enhance the impact of the disease and incidence of the disease. So these are the favorable condition for this disease. The next one is management. In management, it is very important to how to manage the disease according to their characters and according to their disease cycle. We have to create a management structure, and these are the few management structure for this disease. First one is resistant variety. Resistant variety is a very brilliant idea to control the disease. For wilt, we can use Gossypium hirsutum and Gossypium barbendens like variety, like Varalakshmi, Vijay Pratap, Jayadhar, Jariya, Jyoti, G2 and Viram also. We can use these varieties to suppress the disease, to control the disease. And also we can treat the seed with acid-aligned seed with carboxin or chlorothalonil at 4 gram per kg of carbendazim at the rate 2 gram per kg of seed. We can also remove and burn the infected plant debris in the soil after deep summer plowing during June and July, because this disease also come in the field by the infected plant debris. So we have to remove the plant debris from the field. Also, this disease was increased because of a heavy dose of nitrogens and potash fertilizer. If we will uh, balance the dose of the nitrogen and phosphatic fertilizer, it will suppress the disease. And also if we'll increase the doses of potash, this measure also help to control the disease. The next management practices are, we can also apply heavy dose of farmyard manure or other organic manures, follow mixed cropping or with non-host plant. So in cultural practice, this practice will help to control the disease. We have to apply heavy doses of FYM and organic manure, and we can also uh, do mixed cropping with non-host crops, so it can suppress the impact of the disease in the field. And also spot drenching with carbendazim 1 gram per liter of water will also help to control the disease. And in biocontrol agent, we can use trichoderma viridi. Multiply the trichoderma viridi 2 kg in the 50 kg of farmyard manure for 15 days and then apply to the soil, which will increase the trichoderma viridi population, and it will help to the suppress the Fusarium oxysporum F species vesiculentum. So this was the management practice for the wilt. And next disease is anthracnose. So, anthracnose caused by Coletrotrichum gossypi, and this pathogen infect the seedling and produce small reddish circular on the cotyledons and primary leaves. As you can see, there is a in this leaf, in this image, the lesions are small reddish in color and their shape in circular size. So the pathogen infect the seedling and produce small reddish circular spot on the cotyledons and primary leaves. The lesion develop on the collar region stem may be griddle, causing seedling to the wilt, and they will eventually die. In mature plant, the fungus attack the stem, leading to stem splitting and shading of bark. Then the most common symptom is ball spotting, small water-soaked circular reddish brown depressed spot appear on the balls. The lint is tend to yellow or brown, become a solid brittle mass of fiber, and the infected ball cease to grow and burst and dry up prematurely. These was the important symptoms for this disease. Next one is etiology, means pathogen. The pathogen forms acervuli on the infected part, conidiophore and conidia NCT. This is the first one, the first image, which is A image. This one is acervuli or weak, and the second one is city. These C, D, E, these are the conidia and conidiophores. So the conidiophore are slightly curved, short, and the club shape, as you can see in this image. They are developing in this image. They are developing. So the conidiophore are slightly curved, short, and club shaped, as you can see in this E diagram, and their size around 10 micrometer. The conidia are hyaline, falcate, born single on the conidiophore. You can see these are the conidia which are born onto the conidiophore, and their size also lies between 10 micrometer. The numerous black colored thick wall city also produce in acervulus, and this is the city, and this one is the acervulus. So pathogen produce acervulus and city, and inside of in this fruiting structure they produce conidia and conidiophore. Next one is disease cycle. The pathogen survive as a dormant mycelium in the seed or as conidia on the surface of the seed for about a year. The pathogen also perpetuate on the rotten balls and the other plant debris in the soil. Means this disease is also a seed borne और also a soil borne. So जो primary infection होगा वो आपके seed के थ्रू भी हो सकता है, soil के थ्रू भी हो सकता है, जिसमें कोनिडिया आपके सीड में भी प्रेजेंट हो सकती है, और साथ ही साथ इसमें कोनिडिया आपके प्लांट डेब्रिस में सॉइल में प्रेजेंट होने के कारण यहाँ पे आपका प्राइमरी इन्फेक्शन स्टार्ट हो रहा है. लेकिन जो आपका सेकेंडरी स्प्रेड है ये एयर बॉर्न कोनिडिया के थ्रू होता है. सो आपका प्राइमरी इन्फेक्शन सीड बॉर्न एंड सॉइल बॉर्न होगा, लेकिन सेकेंडरी स्प्रेड इज एयर बॉर्न कोनिडिया. दिस पैथोजन कैन ऑल्सो सर्वाइव इन डिफरेंट वीड होस्ट. These are following, and for this disease, for this pathogen, the favorable conditions are prolonged rainfall at the time of ball formation, and close planting is the very favorable condition for the disease, which provide their development because of this condition. The next one is management. How to manage this disease? To manage this disease, we have to treat the delinted seed with carbendazim or carboxyn or thyram or captain at the rate 2 gram per kg of seed. We can use any of these chemical to for the seed treatment. Then We can also we can also remove the plant debris from the soil, which is also a source of primary inoculum. So remove and burn the infected plant debris and balls in the soil. Row out the wheat oats. And in chemical management, for spraying purpose, we can use uh, mancozeb and copper oxychloride or carbendazim also. Uh, at the time of ball formation, we can use mancozeb 2 kg or copper oxychloride 2.5 kg or carbendazim 500 gram per hectare. These these fungicide leads to the suppression for this disease, or they will suppress the inoculum of the disease. So these if we use these following methods measures, so it will suppress the infection of the disease. Next one is bacterial blight or angular leaf spot or black arm. This disease is caused by bacteria sorry this disease is caused by bacteria, and that is Xanthomonas campestris PV malvae serum. So as economic importance, this disease was first observed in Tamil Nadu in 1918, and this disease is very important in Maharashtra, Karnataka, AP, Tamil Nadu, Madhya Pradesh, which is uh, cotton cultivated state. And this bacteria can affect the all stage of the plant, and usually five common symptoms are noticed in this plant due to this bacteria. First one is seedling blight, second one is angular leaf, third is vein blight or vein necrosis or black vein, fourth is black arm, and fifth is square rot or ball rot. these symptoms are ये सिम्टम्स इनके नाम के अकॉर्डिंग ही इन सिम्टम्स को भी इनके सिम्टम्स के अकॉर्डिंग ही इनका नाम रखा गया है। जैसे जैसे प्लांट में सिम्टम्स आते गए हैं वैसे वैसे उनको सिम्टम्स को नाम देता गया। जैसे कि फर्स्ट है आपका सिलिंग ब्लाइट। सो एज यू कैन सी इन द इमेज, स्मॉल वाटर सोक्ड सर्कुलर irregular lesion develop on the cotyledons, and the infection spread to the stem through petioles and cause withering and death of seedling. This is the seedling blight. And next one is angular leaf spot. In angular leaf spot, small dark green color water soaked area develop on lower surface of the leaves, and they enlarge gradually and then become angular when restricted by veins and veinlets, and spots are visible on both of the surface of the leaf. As the lesion become older, they turn to reddish brown color, and infected spread to veins and veinlets too. Next one is vein blight or vein necrosis or black vein. As you can see, the infection is also occurs in the veins of the leaf. The infection of vein cause blackening of the veins and veinlets, gives a typical blighting appearance. On the lower surface of the leaf, bacterial oozes are formed as scales or crusts. The affected leaves become crinkled and twisted inwards and show withering. The infection also spreads from veins to petioles and cause blighting, leading to the defoliation because of this symptom. So these was the vein blighting or vein necrosis. Next one is black arm. As you can see in this image, why it is called as black arm. On the stem and fruiting branches, dark brown to black lesions are formed, which may girdle the stem and branches to cause premature dropping of the leaf. Cracking of stem and gummosis resulting in breaking of the stem, and hang typically as dry black twig to give a characteristic black arm symptom. That's why it is called black arm, because uh, their main characteristic symptom is this typical dry black twigs. So next one is square rot or ball rot. So you can see in this image the ball is rotted due to the pathogen. On the balls, water-soaked lesions appear and turn into black and sunken irregular spot, as you can see in this image. The infection slowly spread to entire ball and shading occurs. The infection on mature balls lead to premature bursting. The bacterium spread inside the ball and lint gets stained yellow because of bacterial ooze and lose its appearance and market values. The pathogen also infect the seed and cause reduction in the size and the viability of the seed. So due to this disease, the balls become rot. Eventually, they will lead to the less yield or distortedly yield. So these was the symptom for the this disease. Next is pathogen or etiology. So sorry. So pathogen as the disease, the bacteria is a short rod with a single polar flagella, and it is a gram negative non-spore forming and measures about around 120 to 1.2 into 0.7 to 0.9 micrometer. The bacteria is aerobic, capsule forming and produce yellow colony in the culture media. As you can see, the yellow color colonies form in the culture media, and these are the bacteria. This is the microscopic view of this Xanthomonas bacteria, which are short rod. The next one is disease cycle of this pathogen. So as you can see this image, this is the disease cycle for this pathogen. This disease is also cause the infection by infected seed, or it is also survive in plant debris. So it can cause primary infection by both, by infected seed also, or by plant debris, infected plant debris also. So this bacterium survives on the infected dried plant debris in the soil for several years. It can be survive for several years. The bacteria is also seed borne, as you can see in this disease cycle image, and remains in the form of slimy mass of the fuzz of seed coat. It multiplies soon after the seed is sown and infect the seedling through the micropyle, and the symptoms will start after its infection. Volunteer plant that arise from the balls falling off prematurely also provide a source of primary infection. The bacteria also attack other hosts like Turberia species, Urodentron species, and Jatropha species. The primary infections start mainly from seed-borne bacteria, and the secondary spread of the bacteria may be through wind-blown rain splash, irrigation water, insects, and other implements which is used in the field. The bacterium enters through the natural opening or insect caused wounds. So these were the disease cycle for this disease, this pathogen. The next one is favorable condition. So this disease is favored by optimum soil temperature of 28 degrees Celsius, high atmospheric temperature of 30 to 40 degrees Celsius, relative humidity of 85%, and early sowing, delayed thinning, poor tillage, late irrigation, potassium deficiency in soil, and rain followed by bright sunshine during the months of October and November are highly favorable. So these was the favorable condition for this pathogen which favors to development of the bacteria. Next one is management. To manage this disease, we can use multiple management practices. So here are some. Remove and destroy the infected plant debris. Rogue out the volunteer cotton plant and weed host. Follow crop rotation with non-host crops. Early thinning, good tillage, early irrigation, early earthing up, and addition of potash to the soil reduce disease incidence. Resistant variety like HZ9, BGA 592, G27 Sujata 1412 and CRH 71, and these are the resistant variety. There is a one tolerant variety also, which is Suvin. We can use this resistant and tolerant variety to escape the disease. So next one is, we can also use this following measures, like dilute the cotton seeds with concentrated sulfuric acid at 125 ml per kg of seed. Next is, treat the diluted seeds with carboxin at 2 gram per kg seeds, or soak the seed in 1000 ppm streptomycin sulfate overnight, or treat the seed with hot water at 50 to 56 degrees Celsius for 10 to 15 minutes. Sorry, it is looking 0 after 6, but it is 56 degrees Celsius. So the water temperature should be 52 to 56 degrees Celsius, and we should treat the seed for 10 to 15 minutes only. Next and last one is, it is a bioagent also, and this is a chemical compound also. Spray the streptomycin sulfate, that is also called as agrimycin 100, 500 ppm, along with copper oxychloride at the rate 0.3%. These following measurement definitely will suppress the disease in the suppress the incidence of the disease in the field. So we should use this for this mentioned management practice